So students, we were discussing about the elongation process. So how many steps are there in case of elongation? There are 3 steps. So the first, that is codon recognition. The first step, that is codon recognition. Now what is this codon recognition? I will make the diagram again. Like this is the small ribosomal subunit and here the mRNA, and it is bind with the AUG codon. Means the tRNA, it will bind with the AUG codon having the U, A and C. And what is present here? Methionine. Okay, this is present in the P site. Now this site, it is empty, and what is the site here? That is A site. Okay, and I told you then that the A site, it is responsible for the charged tRNA. Means the charged tRNA, it will enter in the A site. Okay, so this is the A site, and in this A site, what will happen? The charged tRNA having the C, means the complementary codons for C, C and U. Okay, it will come here. Means in the mRNA, the second codon, this is the first codon and this is the second codon, that is CCU over the mRNA. So on the mRNA, when the codon, that is CCU, then the complementary base sequences, it should be C base pairs with the G, C with G and U with A. Okay, so this should be the complementary codons over the tRNA molecule, and this is the charged tRNA, and this charged tRNA, it will bind with the codons CCU. Okay, and it will enter the A site. So when the codons that are CCU, then the amino acid which will bind with this tRNA molecule, it will be the proline. So I am writing here PRO. Okay, here this is methionine. So the first, that is methionine amino acid, and second I have written proline. Okay, now it will come here, and this side, that is empty, that is the E site, and all this, that is present in the large subunit. All of this, it is present in the large subunit. Okay, so codon recognition means here that when this charged tRNA, it will bind with the CCU codon which is present over the mRNA molecule, Okay, so this is known as the codon recognition. Means that the codon, that is recognized by the charged tRNA molecule. Now the second process, that is the peptide bond formation. Peptide bond formation. Now how this peptide bond formation it occurs? How this peptide bond formation occur? Means like this is the first amino acid and this is the second amino acid. Okay, so this amino acid, it will break from here and it will form a bond with this proline. Okay, if you know that which bond that is present between the two amino acids, yes, this is the peptide bond which is present between the two amino acids. The peptide bond, it is present between two amino acids. Let's discuss the structure of one amino acid, that is C H R NH3 and here COOH. This is the structure of the one amino acid. And if we will take another amino acid, suppose this first amino acid, that is the methionine, okay, then second, that is NH3 and COOH here. This is the second amino acid. Okay. So the peptide bond, it will form the COOH group of the first amino acid and the NH3 group of the second amino acid. The first amino acid having the COOH group and the second amino acid means having the NH3. And the peptide bond, it is represented by CONH. This is the peptide bond. Okay. So this bond, it will be breakdown, means this bond, it will be breakdown from here and it will make the methionine, it will make the peptide bond with the proline. It will make the peptide bond with the proline. Okay. So let's see here. This is the small ribosomal subunit and this is the mRNA here. Then 5' and 3' here. Okay. Next here, that is A, U and G having the codon, and second is C, C, U. And here the charged tRNA, here also one charged tRNA it is present. Okay. This charged tRNA, it is bind with methionine and this is with proline. Okay. So now what will happen? This, it will break from here, this bond, it will get breakdown and it will form a peptide bond with the second amino acid having the proline. Okay. It means that this tRNA, it is without any amino acid. Now this tRNA, it is not having any amino acid, and if suppose the amino acid that is not present with it, so it means that it is discharged. It is the discharged tRNA. Okay. So methionine and proline, they make a bond here which is peptide bond. So it means the peptide bond, it forms between the two amino acid, and then what will happen? This E site, it is again empty here. Okay. And P is containing the it is containing the discharged, means not having the charge. Discharged tRNA, it is empty, and this is containing the 2 amino acid. Okay. So this is known as the peptide bond formation. Now this peptide bond formation, it occurs in the presence of the ribozyme enzyme. It occurs in the presence of the ribozyme enzyme. Okay. So ribozyme enzyme is actually D23sRNA of the ribosome, and this part, it acts as the ribozyme, and it is also known as the peptidyl transferase. It is also known as peptidyl transferase. It is also known as the peptidyl transferase. Okay, so this is all about the peptide bond formation. Now, third, we will discuss about the translocation. Translocation. Now, in the translocation, what happened? In the translocation, what will happen? That this A site, this tRNA, which is present in the A site, it will move to the P site. It will move to the P site with the codons. Okay, means this tRNA, it is binded with the codons. So with the chain, with the mRNA chain, this tRNA, it will move to the P site. It will move to the P site, and this is known as the translocation process. Now see what happens when it will move to the P site. Now this is a small ribosomal subunit, and this is A, U, G, and next is the CCU, CCU, first codon, second codon. Okay, now third we will write here A, U, C. Okay, this is the third codon. Now you can see the difference here, that here the AUG, here the AUG, it is present in the E site. It is present in the site. Okay, and the P site here, it is having the tRNA which is binded with the proline and methionine. Proline and methionine. Okay, and now here this is empty, this is empty. So this is translocation process. Translocation means that this A site, the A site having the tRNA, this tRNA it will translocates in the P site. Okay, so when this tRNA means this having the complementary base pairs with the CCU, that is GGA. It means it is having GGA anticodons. So when this tRNA it will come in the P site. Okay, so the discharged tRNA which is already present in the P site, it will come in the E site. Okay, so it means it is having the discharged tRNA. It is having the discharged tRNA, and E means exit. E means the exit here. So this discharged tRNA, it will be removed from here. It will be removed from here, and that's why it is known as the exit site. And P site having the tRNA which has 2 amino acids. And this A site, it is filled by the another tRNA having the complementary base sequences with these codons, that is A binds with the U with A, and C with G. Okay, so it will code for isoleucine. It will code for isoleucine. So this is the third amino acid. Okay, so when this tRNA it will bind here in the A site, then this is the same process, that is the codon recognition. Okay, so when this tRNA it will bind here, then this is known as codon recognition. Then again peptide bond formation will be there, means this bond it will get break down from here, and this will make the bond with the isoleucine. It will make the bond with the isoleucine. Means the COH group of this proline, it will make the peptide bond with the amino group of the isoleucine, and this is the peptide bond formation. Then again the translocation process occur, means this will shift to the P site. So in this manner this chain it will repeat it again and again, means first codon recognition, then after that peptide bond formation will be there, and then translocation. So one by one the amino acid it will add it to the chain, and it form the polypeptide, means the protein. Okay, so next step, that is the termination of the translation, that how the termination process it occurs.